And I think first one is Steve Shirtliffe, and Steve is an agronomist who works in a professor and agronomist who works in plant sciences here, and he's been leading the field phenotyping program, both from the point of view of overhead imaging using AUVs, and also now with the various phenomobiles that we're starting to work with this year. So Steve, if you'd like to come up.
Thanks very much, Morris. Let's give Morris a hand for that great introduction. Okay, just come in and have a seat. Don't worry about disturbing me. I'm used to lecturing, so if there's a lot of buzz around there, that's kind of something I'm used to. Okay, so let's if we could have, do I just hit the button here to get my talk up? There we go. Okay, so I'm going to talk about field phenotyping of crops with unmanned aerial vehicles, UAVs today. And but first, I want to talk a little bit about my co-authors. There's the usual gang of people in my own lab. You have your postdocs like Hema and graduate students and technicians, Meng Lu. But the key thing about this PERC project, which I can't underemphasize, is the degree of collaboration with it. We soon get past my graduate students, and we get into plant breeders, Kirsten Bett, Sally Vale, and Curtis Posniak are in there. So we have plant breeders in 3 major crops: lentils, canola, and wheat. And we also have a bunch of other people in computer science and engineering working with us, like Kevin Stanley, Ian Stavnis, Mark Aramian. And then I kind of ran out of space, and I knew I'd forget people, so I put Carl Gutwin and company, and all the computer science is in his field. I know we've met with many people over the couple years we've been running, and Scott and his thing. For whatever reason, there's a bunch of Kevins and a bunch of Steves in this project. So I think if we're going to phenotype phenotypers, we'd look for people by the names of Kevin and Steve, and that would start us off. But anyways, we'll start there. Morris gave a great introduction into phenotyping and what is the overall motivation. I want to talk a bit about the specifics, and of course, if you don't know what it is, I borrowed this slide from one of my graduate students, Karsten Nielsen, and it's assessing the physical and biochemical characteristics of a plant in its environment. So it has both it's from the genetics of the plant, but also the environment of the plant interacts to express it. And it's essential for breeders for variety development, because that's what they select on. One of the key, of course, yield isn't what breeders always select on. They're looking for other phenotypes as well. And yield's a very difficult one, because you could argue probably every gene in the plant goes somehow into controlling yield. So you'll select on indirect methods, but there's 2 reasons you use it. There's variety development, just the mechanics of somebody running a breeding program, selecting, I don't like, a breeder doesn't want anything that's too tall because it may fall down. So they only select shorter varieties. So they go through and make notes on that. And there's also the aspect that Morris talked about, this idea, it's not an idea, we know that regions of the genome control expressed phenotypic traits. The trouble is that we can't phenotype that well. There's challenges with the traditional phenotyping. We're often giving qualitative ratings, or often given to quantitative traits. If you look at I'll see if, can you see me touching on this, or? No. Oh yeah. Ooh, I just drew something weird on there. On this, or did that show up on there?
I can do it. You can actually, if you want to use the pad.
Oh, okay. Maybe we'll just, if you look at those 3 p-plots in the front, if you were to pick the middle one, you might say, oh, maybe that's shorter or something, or maybe a person might take a note of, marginal canopy development, but the one to the right is good canopy development. Well, what the heck does that mean, marginal or good? How do you do stats on that? How do you associate something like that with a region of the genome? So it's not thing. In addition, there's also I don't need to see those marks anymore. You can just make them go away and go back to the regular one. Yeah, thank you. My little squiggles in there. And there's also, as Karsten also put, as a person who's gone out and worked in both private industry and academia, he used a lot of negative adjectives. It's monotonous, slow, expensive, destructive, labor-intensive. And those of you that know Karsten, I can see some people smiling, know that he has that Scandinavian kind of way of relishing in what is what is difficult, so that there. And of course, the final one, which has been brought up, there's questionable consistency and objectivity between observers and over time. And we'll look at a little bit of this. So let's just start here. We have a little video here we'll watch. And I want to, there's a canola, if we could click the video to start it, please. There we go. So this was some cameras that were put up by Kevin Stanley and Ian Stavness in our plots. These are actually just originally just a security cams. What you're seeing is canola growing. And every time it gets a little bit dark, that's one day that goes by. What is extraordinary about crop growth here in Western Canada is how quickly it happens. This dimension of time is something we don't often consider enough in. We're already here. You can actually see leaves individually expanding in these pictures, right? You can kind of, it's kind of slowed down a bit now, but you can, within a day, you can see basically one leaf there. You can see one leaf has completely come out of the plant within a day. And, within about 10 days' time since this video started, it's gone off from maybe 40% ground cover to complete. Now we have flowering coming, within a day, we're seeing extension of the primary rachis. You wait a couple more days, and now you see the secondary ones, the branching ones coming up there and filling the space. These are all phenotypes that if you're out there marching around with a clipboard trying to rate, let's say, flowering in a plant, in a crop, how are you going to rate them, and is it going to be important? And can you actually rate the timing of it? Well, no, you can't really at all. But anyway, so we'll move on to that. So that's just some more motivations about it. As Mark said, we're going to concentrate on UAVs and on ground-based vehicles. We've started with some there, and with UAVs, you can get decent resolution. I'm going to show you some infrastructure that we've recently started using where we're actually getting really good infrastructure. And there's also ground-based vehicles, which Scott Noble's working on. We're going to focus today mostly on UAVs, but a bit on ground-based vehicles, but on our early generation ones, which I'll show you, the ones that before we had engineers involved with the project that we developed. So Morris showed you this slide here. We've started off partnering with a local company, Dragonfly. All of the success that we've had in early imaging can be traced back to our corporate involvement with Dragonfly, that we had a partnership with them, that we had somebody right in Saskatoon that could integrate sensors on a UAV, and when we had a problem, which was really rare, go there and get them fixed. So we worked with them over time. We've started off simple with an RGB sensor, that Sony RX100 up there, just a simple high-end consumer grade. We soon switched to a multi-spectral imager there. You can see it has 5 little lenses on it because it has 5 little cheap cameras essentially on it to capture imagery, but it captures them in distinct bands. We'll get into more of that. And we've also looked at converted cameras, as well as in the bottom right-hand corner, you can see a little thermal camera. I'm not going to be talking much about it today. In the past couple of years, we haven't had a good opportunities to do a lot of thermal work because it hasn't been that hot and dry. Looks like we may be doing some thermal work this year, though, I would suspect, because it's that kind of year. The multispectrals, if you look at that graph in the top right, what they give you is distinct bands in some key wavelengths that we're interested in. They're good so they give us reasonable spectral resolution. We're seeing the spectra of the plant across there. If you're familiar with our NDVI, I can't really point on this. But anyways, you can see the spectra, but we're seeing the blue, the green, and the red, and then the red edge and the near-infrared. Near-infrared, of course, is important, the near-infrared, because vegetation reflects a lot of it, and that can be kind of as a baseline for a lot of stuff. We can use other cameras as well that give us better spatial resolution. The issue being there, though, of course, is that, as you can see, we're getting some overlap with them. We've since moved to a system this year. We got some Western economic diversification funding generously that has allowed us to step up our platforms, and we've moved to we've fallen in and started buying, which is like the company that is like the Henry Ford of drones. They may not build the best ones, but they build them really cheap, and they're always black. And they build the most is the DJI. Dragonfly is still partnering with us. What they've done is it's maybe hard to see, but we've actually co-mounted 2 cameras on there. So instead of having to fly for a high-resolution RGB image and then come back later for a multispectral, we can do it all at once, and we have RTK sensors on that, so our position of the drone is within a centimeter. We know where it should be within a centimeter, and we can do very well there. And on this camera, on one of these instances, we've actually mounted, and Morris says we don't mount the $30,000 cameras on a drone. Well, sorry to tell you, but we are. but we did mount a 100 megapixel medium format camera on the left there, and multispectral cameras. I wish I would have brought some imagery from the new camera, but it is from 30 meters high. We can see lentil leaflets. It's incredible. And Andrew from Dragonfly's back there. He asked me once, Steve, what do you need so much resolution for? Are you trying to count the spots on the back of a ladybug? You know, it's like, well, no, and it isn't that good. But what we need it for, and we'll get into that in a second when we talk about what's being done with some of the computer sciences and some of the advanced image analysis. Why we want really good spatial resolution. Here's a video of it flying. The first one, if you could click the video there, please. This is our second flight with it, about a week and a half ago, or I guess about a week ago, and there it is. It weighs about 40 pounds, and it takes off and flies up in the air and takes a bunch of pictures. And people often ask, is it fun to fly UAVs? This one, you're flying it. Hema, the guy with the looking up in the air, he's flown almost all of our missions. He's the unsung hero in this. Not to take anything away from him, but flying these new ones, there's 3 buttons to press. Take off, and then you got to press execute flight plan, and then there's return and land. And that's all you're doing, unless something goes wrong. So it really isn't that difficult. We're also getting into hyperspectral imagery. We have Keshav Singh is here. He's working on using hyperspectral imagery. We have a hyperspectral camera that we're going to be flying with our UAV there. We've done work with Scott Noble as well. Scott Noble's a hyperspectral. He did that for his PhD and MSc, I believe, so he's got a lot of experience in the lab-based part of it. And with a hyperspectral imagery, what you get, instead of just having like either 3 bands or 5 bands across the spectra, is in this case you have 155 bands. So you have essentially what they call a data cube. So instead of like an RGB layer of red, green, and blue with the spatial information within that, you have 155 bands depth of that. So you essentially have this huge amount of data in this data cube that you can tease apart. We don't think that we're going to use this for everyday stuff, but we're going to use this as a research tool to figure out what individual spectra are important in this and do that. So anyway, Morris and I are sharing some of the plots, and he taught some of the slides, so we'll talk about it. We should have talked about that, but I'll focus on some of the analysis. This is some work done by a student of Mark Aramian's, where he used image analysis, in this case textual analysis, classic image analysis, and to look at lodging in wheat. And that we really wanted to look at stay green in it that year, but we had this lodging which was messing it up, so we thought, well, if it's raining lemons, let's make lemonade. And that's been kind of maybe a theme of this part of the project, is that we've been we've been advantageously phenotyping what expresses itself, and so there. They've also done some work with Jason Ubens. They started some work with our computer science, Ian Stavnis' student, Jason, did some work first on an existing image data set on using deep learning. Probably many of you have heard of deep learning. It's one of the most exciting scientific tools that's recently emerged. It's an artificial intelligence type of thing. And in deep learning, what happens is you have these convolutional layers where you have these smaller, essentially, kind of filters that scan the image. If you can think of it as a big scanner going across it, and those 11 pixel by 11 pixel boxes, and working out different mathematical relationships as it moves across each individual pixel. It can then build a matrix of relationships using different filters and different levels, and the computer itself actually learns, uses different filters, and essentially tries to figure out a way of training an image to what you're trying to do. In this case, somebody would have gone in and manually counted how many leaves in a training data set, and then the computer would have gone out and they would have fed it into the program so the computer could learn what image traits make it a leaf. And you can see those two little below the filters thing there. You can see it's probably looking at the leaf edges and using that as some way of counting leaves. But the actual operator, the programmer, isn't actually saying, do this, if thing looks like, if the geometry is close to being round, then do this. They don't program that in. It just happens automatically beneath that. So they were able to do this, and they published this paper in Frontiers. In terms of the field stuff, we've collaborated with them, and IH just published a paper in a referee conference proceedings a few months ago. I just got a notice while I was sitting here that it's already had 200 reads on ResearchGate, and it just came up a few months ago on using this to count individual plants growing in the field, which is something that sounds trivial, but is actually very important as kind of a housekeeping type of thing, because if you have different amounts of plants in different plots, your results can vary. So you can using this, you can correct a lot of your results. So that's been interesting. We're using this imagery, and this is the first ground-based platform that I developed. Went over to Co-op and bought $200 of the lumber and build something. It was. I remember putting in the claim. I think it was $216 of lumber. It was before GIFS was around. Before PERC was around, and we started imaging wheat heads in it and trying to use that method. Oops, go back one. And using that method to. Oh, I keep pushing. The laser does. Oh, here, I can use the laser on this. So we're manually. So to make this method work, you have to annotate. You have to have somebody that annotates, which means essentially put dots on a training set, and then the computer has to learn that, puts this through those filters. Of course, with wheat heads, we're running into problems of occlusion, which means that individual wheat heads are blocking each other out. What Nick is looking at, which is the grad student you saw driving it there, is a lot of what is basically comes down to good photographic procedures. What are the best situations? What's the best timing to do this to get the best estimates? So we're going out and counting all the wheat heads in individual plots and trying to figure out a way of, one, being able to capture an image that we can see most of them in, and two, to be able to train in collaboration with Ian Stavness and some of his students to train a deep learning methodology to be able to count those in the field. T. Zhang has been working in the canola, and you can see canola flowering there over time. We're using more classical methods in this case. We're either looking at the yellowness of the plots by just using some multispectral information, like green minus blue, as it gives you a good indication of how yellow something is if you subtract those bands, and stabilize it. And also as well, we're actually extracting the pixels. I'm acting like I'm a neophyte up here. Extracting the pixels from that above a threshold value, which are canola ones. And we're going to be working to actually try to find those in there with image analysis as well. But for now, that's what we're doing. We're getting reasonable relationships. You can see on the top left-hand graph of the yellowness versus actually the flower number. They're going out and counting individual flowers on canola. You talk about phenotyping you don't want to have to do. That's a difficult one. So we're getting good relationships with that and with the thresholded index on the right-hand side with different genotypes. And the exciting thing is when we look at this over time, is we can actually calculate how much a plant is flowering, but we can also kind of calculate the volume of how essentially the volume of flowering over time. If you're to look at the bright yellow, at the highest peak in there, the bright yellow one, you see that it's kind of a let's get her over with kind of canola plant. It flowers hard, flowers early, and then stops flowering. Whereas you have others that are more of a, this is flowering, so I'm going to try to avoid any references here that may be inappropriate, but that last longer over time. So these are new phenotypes here. You could go out and rate flowering before, but here now we're able to rate flowering at given times. We're also able to calculate the duration of flowering, and by multiplying those by each other, essentially get a kind of a flowering energy. If you think of like kind of power times, kilowatt hours, the equivalent of something like kilowatt hours, it would be over that. I want you to call this a flower power phenotype. She's not crazy about that, but I think it would be interesting. The plot on the right-hand side is showing not a good relationship with yield, which is what we expect. Because just because a plant sets a flower, it doesn't mean it's going to produce the same amount of seed. The seed size, how many seeds per pod are in there, are all going to come in there. So, of course, there's other yield components that come in there, but we do have a reasonable relationship. But that being said, phenotyping lentil, if we could click this one, we'll talk about lentil phenotyping. We'll end on, this is our final crop we'll end on. Lentils are a wonderful crop, but they're a challenging crop because they're small, they're short, and the canopy takes a long time to fill in. You can see here, it's been going a while, the canopy, and some of these ones was kind of thin to start with, and now it's finally filled in that one there. And of course, that's what we want. We want a canopy, a plant canopy. Essentially, the physiological mechanism of it is to capture light energy and then to convert it into energy for the seed to actually have harvestable material. So the sooner we can have a plant canopy, and then of course, we didn't spray these plots. We're seeing differential expansion of lentil disease in these ones. So they start to die off from disease on the other side. So we want a lentil plant that can grow early and fill in space. And in fact, I believe the original lentils that were released in Saskatchewan were selections from the World Collection that were essentially visually selected for height, essentially, so we could mechanically harvest them, and for standing up, that those original ones were. So we're going to backtrack a little bit here. Now I'm going to talk a little bit about fababine. And the reason I'm talking about fababine, because this is a trial we did for herbicide tolerance in fababine. I did with Chris Willenberg and Eric Johnson. And this was just done as a little side project for an undergrad project. And we thought, hey, wouldn't it be cool to see if we can tell if we could actually measure how tolerant plants are to different herbicides based on using our imagery method. So we went in there and we segmented out individual rows because we had problems with weeds still being in some of the plots, so we had to just do a little bit of manual imaging, manual tweaking in there and a bit of thresholding to get it out. But when we did get it out, we compared it to some ground truth ratings by a friend of mine, Eric Johnson, who's up in the right-hand corner. And Eric has been doing herbicide trials since for a long, he's not that much older than me, but he's a bit older. He's been doing them for a long time, and he's really good at them, and he's a really honest guy. These are Eric's writings on the left-hand side in visual chlorosis. And you can see, I'm not going to go into it, it's all the same lentil genotype, but there are different herbicides causing different amounts of damage in the plant. And what you'll notice there is there's that pattern of kind of how the herbicides cause the damage. We won't get into details about that. When we go over to the right-hand side, those are based upon our UAV, just a relatively simple workflow of just using indexes and how many pixels above a certain threshold and how tight our data is for those. What we're being able to see is that we're able to tell differences between individual treatments that in the other one, because of user error, are not that you can't tell at all. And in fact, our CVs, our coefficients of variations, were about a third of what Eric's were. And not to say anything bad about Eric. He's really good at it. This is just a better methodology for doing it. And there's a second year as well, showing that it wasn't just a fluke. This work, of course, has been realized before. Vlad Pajic, who's a graduate student with Kirsten Bett and is now working with Bert Vandenberg, first proposed to use this on lentil to look for tolerance to do different herbicides. And in fact, we're continuing that work with Bert and Vlad, looking at it to try to find some tolerance to these herbicides. And they're looking for natural tolerance. They're not looking for, there's no gene, they're not inserting a GM gene into that. So they're looking for natural. So they're looking for probably in many cases what will be small differences and to be able to link those to the genome, or to be able to tell those differently and to be able to actually move those through the selection process. And in fact, Vlad did some work on this on his PhD. Some of his work here you can see in that middle set of, that middle row of ones, you can see some plots that look, those dots look a bit hotter, indicating that they weren't hurt as much by the herbicide. So this is a good methodology for that. And in fact, we've been out this year already doing some ground and aerial-based measurements. We're on a really windy day, so we went out. I think there's a little video here. This is our redneck phenomobile that this is what happens when agronomists build things and not engineers, as they look like this. Anyway, so the last thing I'll talk about is some work we're doing with Karsten Nielsen on using imagery to estimate lentil canopy growth. Again, related to that same issue of lentil not being the world's most vigorous plant and smaller, is trying to find ways to phenotype traits that may be associated with that. We're looking at both a two-dimensional approach and a three-dimensional approach to do this. And I'll get right into this. In terms of germplasm, I haven't talked as much about germplasm as I should have in this. In T's line, in T's experiment, we're looking at the 50 NAM parental lines. So these represent a very diverse set of germplasm that has some genetics, that has a genetic structure that's been recently characterized that we'll be able to take it further on. But in the lentil one, we're looking at the agile panel, and that stands for something. I'm not, you can read it if you want. But anyway, so it's a diversity panel, this is I just wanted the first one to come up. So there's the large experiment that has 324 lines. We've imaged them in 2 locations in Saskatchewan for a couple years, and Carson went over to Italy and did some imaging over there as well. So we have a true international. It actually also exists in Africa as well. This project is pretty incredible. It's on 4 different continents, I believe. Or is it 3 or 4? Anyways, it keeps double bumping this. But we're using some training data, and on the upper right-hand corner in blue, this is the data I'm going to be presenting, where we've actually gone out with 6 diverse varieties from this panel and harvested the biomass and are relating that back to the imagery. Of that. So we have, for the two-dimensional growth, we're able to do that by just pulling the plots out and getting an idea of how much ground cover is in there. If you can see that there's essentially looking at how much green area is in there. There's a thresholded version, so just calculating how many pixels are covered in that. And that's pretty exciting. We're able to get, early on the relationship isn't good, but if you look in the bottom row when we get later, we're getting a nice relationship between green pixel area and crop biomass. Of course, it changes over time. If we look at the slope of those graphs, if we get down to some idea, is they actually change over time. The slope increases because the slope is growing in height and growing in biomass over time. So we got some parameters there that are changing over time, but we have a good relationship over that. Oops, back. Now, if we could click the video here, Morris talked about how when we stitch together this imagery that we put together a lot of images. When you take 2 images from a moving vehicle, you get a slightly different perspective. That allows you to reconstruct a three-dimensional map, a three-dimensional map of a point cloud of what the imagery would look like. Because, just like a 3D camera, you have two lenses. When we're flying the drone, every place in the plot is getting at least 5 images. So those differences in geometry, we're able to reconstruct with the software a three-dimensional view of what it's like. That movie of the moving around there is not from the drone moving around. All those pictures were taken with the camera pointed straight down with the drone flying a grid pattern. We're just moving around to be able to see, so we can see what that canopy is like. Well, when I first saw this, I was really excited because I realized we could start to actually look at canopy volume with this. And we stole some ideas from the remote sensors in the tree world who had done this kind of work in trees already to calculate the volume so they can see how many board feet they have. And so we're able to do that within the plots. And here's Karsten's biomass plots here. And you can see, so essentially the green area around that little lentil plot is the ground level, and everything that's red is above. So essentially we're kind of digitally slicing off the biomass, and we're able to calculate the volume there. In that green square in the bottom left-hand corner, you can see that the volume is 0.18 meters squared of volume there. Well, if we look at that over time, we can see that initially we're not getting a great relation because there's too much. We're trying to measure lentils that are an inch tall. Excuse me for using English units. But as later on, we're getting a really nice relationship between volume and dry weight. And again, these are in very diverse lentil genotypes. What's interesting, and maybe I should mention another thing there. What's also interesting about this, if you start to think about some of the things that are here, is we're actually on the y-axis, we have dry weight, and it's by volume here. So essentially, the slope of that line is giving us the density of the lentil canopy at that time. So we're able to calculate that, and we see our density of the lentil canopy changing over time as that slope changes. Again, some more potential phenotypes. If we take this home to the last slide here, if we look at crop biomass, of course, anybody who's been involved in plants will know that the plants have that characteristic biomass accumulation. The two-dimensional area graph that we can see in the middle panel, we see that, of course, the plants cover that area on a 2 dimensions much quicker. Well, finally, I don't know, it always keeps double jumping here. Finally, the last one, the 3-dimensional volume, is a little bit different because it actually goes up, and you see it actually starts to decrease after a little while. But it's also, but it has some characteristics there that now, because we have a time measure in here, we can characterize these individual genotypes. We can use nonlinear regression to calculate how long did it take them to reach maximum volume? What is the rate of change of maximum volume? So we're essentially discovering new phenotypes that we think may be important for plant phenotyping. So that's kind of the sum up of the 3 crops that we've done: the flowering intensity, the lodging and yield components, and herbicide tolerance and canopy accumulation in lentil. We're working on a lot of others. The next step of this is using these phenotypes for selection criteria in breeding programs, and of course, genomic association. We're looking forward to doing that. We've started it a bit already, and some of the genetic tools are coming into place, so it should be a lot of fun. That's our team in my lab that does, of course, there's a lot of people, and these are some of the people that have paid for parts of this. Thanks very much.